Hey everyone, welcome back to Journal Club 2. This week we'll be discussing the article Exercise Promotes Healthy Aging in Skeletal Muscles. This article was created by Gregory Carty, Russell Hempel, Marcus Banham, and Julian Zyrath, and it will be presented by me, Rachel. So the article kicks off by describing primary and secondary aging. So primary aging is the unavoidable deterioration of cellular structure and biological function. This deterioration is independent of disease, lifestyle, and environmental factors. There is no way to prevent primary aging. It happens to everyone that gets old. Now, secondary aging is the decline in structural and functional changes due to aging. This time, it can be caused by disease, lifestyle, and environmental factors. We will touch back on secondary aging in a moment. I just want to mention that the article then continues to differentiate physical activity from exercise. Just to review, physical activity is any level of skeletal muscle activity that results in movement, whereas exercise is planned, structured, and repetitive. Exercise is typically aimed at improving physical fitness. The article then went further to define endurance and resistance exercise. However, I will assume that everyone will know the difference between the two of these based on your extensive kinesiology education. Alright, so now let's go back to secondary signs of aging. This article discussed warding off secondary aging. One of the greatest contributors to secondary aging is physical inactivity. Physical inactivity increases the risk for type 2 diabetes and increases the risk for metabolic syndrome. An increase in physical inactivity will also lead to a decrease in muscle mass. This article chooses to use the term aging muscle atrophy. It chooses to use this term over sarcopenia, which is the age-related loss of flesh, because it incorporates cellular changes, a decrease in mobility, and muscular performance. Since physical inactivity promotes secondary aging, increasing physical activity will reduce and in some individuals prevent secondary aging. In adults 70 years and older, any physical activity will decrease the risk of type 2 diabetes, and in adults 50 to 69, moderate to vigorous physical activity will decrease the risk of type 2 diabetes. Now, since we're discussing type 2 diabetes, let's look at how insulin sensitivity changes with age. This article describes the insulin signaling pathway for glucose transport. I'm going to walk you through the signal cascade. All the steps will be along the bottom for you to follow along. I'm choosing to use the acronyms instead of the full names because I'm finding them very difficult to pronounce, and it's not something I want to spend time stumbling through. So let's begin with a cell membrane and an insulin receptor. Along will come insulin and bind to the receptor. The receptor will autophosphorylate. IRS will also phosphorylate. IRS, once phosphorylated, will then bind to PI3K. PI3K will then phosphorylate AKT2. AKT2 will then phosphorylate TBC1D4 on the serine and threonine photomotifs. This will then translocate GLUT4 to the cell membrane to transfer glucose into the cell. So this is the normal, fully functioning signal cascade. But with age, this signaling process can become defective. This occurs when skeletal muscles become insulin resistant. Thus, the signaling pathway is not happening and GLUT4 isn't translocated to uptake glucose. However, exercise can counteract these changes in age-related insulin sensitivity. Let's begin by looking at acute exercise. Acute exercise will lead to an increase in insulin-stimulated glucose uptake due to GLUT4 translocation. This is a signaling pathway that we just discussed. This increase in insulin activity can occur 1 to 7 hours after exercise and could potentially last 1 to 2 days. Even a brief 1-hour brisk walk can trigger this response. Then we move on to chronic endurance exercise, which leads to an increase in glucose disposal, an increase in TBC1D4 serine and threonine, which was the last step of the signal cascade that we just saw, an increase in glycogen concentration, and an increase in capillarization. There are similar benefits with chronic resistance exercises. Again, there is increased glucose uptake. There is also an increase in that final step, except only theanine will phosphorylate. And there's also an increase in muscle mass. Thus, both endurance and resistance training is important for maintaining insulin sensitivity. The article moves on to discuss mitochondria and aging skeletal muscles. So age-related changes in mitochondria impact skeletal muscle mass and function. Mitochondria and skeletal muscles are highly adaptable. Mitochondria content will fluctuate depending on the metabolic demands of the muscle tissue. Thus, physical activity is a determinant of mitochondrial content. Some muscles will see an increase in mitochondria, and others will see a decrease. These changes in mitochondria content occur at different points of the aging spectrum and are independent on the muscle fiber type and activity. So each tissue might look a bit different. Some muscles contain severely atrophied, deinnervated muscle fibers that intermingle with normal-looking fibers. This depends on the muscle fiber type and the activity levels of that muscle. I will mention later that atrophy is more common in type 2 fibers and discuss this concept further. Now let's review mitochondrial function, just in case anyone needs an MBB refresher. So, mitochondria are involved in ATP synthesis, reactive oxygen species signaling, and regulating intrinsic apoptosis. So let's look more into that mitochondria and ROS system. So an increase in ROS is produced by mitochondria due to the aging skeletal muscles. This leads to impaired contractility. It used to be believed that an increase in ROS leads to atrophy. However, this seems to be overemphasized as studies now show that an increase in physical activity in individuals doesn't show an increase in ROS and can actually limit ROS production. The article moves on to discuss mitochondria and apoptosis. So under stressful conditions, mitochondria undergo a permeability transition. So a pore known as the mitochondrial permeability transition pore opens on the membrane surface. Large proteins are able to leave the mitochondria and induce cellular apoptosis. Physical activity can't prevent this pore from opening. Therefore, aging skeletal muscle, changes in respiration, and an increase in ROS can make the cell more prone to cellular events. Mitochondrial plasticity is the next topic that we're going to discuss. So, an increase in physical activity will decrease mitochondrial changes and it will also boost mitochondrial biogenesis. So here's another signaling pathway that we will go through together. So an increase in physical activity will increase the amount of muscular contractions in the body. With an increase in muscular contractions, we see an increase in adenosine monophosphate kinase and SIRT 1 levels. AMPK and SIRT1 will induce peroxisomes proliferator activated receptor gamma 1 alpha isoforms. This will then lead to an increase in biogenesis of mitochondria. Thus, physical activity will increase the cellular pathway that lead to more mitochondria in skeletal muscles. This is particularly true for endurance athletes or endurance exercise. So the older adults will initially benefit from endurance exercise and see an increase in muscle aerobic capacity. However, as they enter the oldest age category, this benefit will be lost and they will see an accelerated decline in aerobic capacity to match that of about sedentary individuals who never went through the physical exercise or the endurance exercise. This shows the mitochondrial plasticity as there is some failure to upregulate PGC1 alpha with advanced age. Now, let's move on to one of the last topics presented in this paper. Age-related muscle atrophy. So muscle loss can lead to functional decline, disability, and dependence. It may also lead to a decrease in mobility, an increase in obesity, and an increase in type 2 diabetes risk. Muscle atrophy is a multifactorial degenerative process. Thus, it has many different influences. As previously mentioned, atrophy occurs in type 2 myofibers. These are the fast twitch, but both type 1 and type 2 fibers can be completely lost. Hallmarks of atrophy are a decrease in neuromuscular performance. So a decrease in strength will speak to the muscle quality. So greater strength is typically lost in the legs rather than the arms. And a decrease in power means that there's less excitation-contraction coupling. Of course, as per the nature of this article, exercise can decrease atrophy that occurs with aging. Resistance training will increase neuromuscular adaptations because of hypertrophy. This will increase power and strength. Notice that there's no hyperplasia. This is because the cells are terminally differentiated. Thus, they can only increase in size and not in number. Also, endurance training will increase insulin sensitivity, which is that first signal cascade we discussed, and it will also decrease the amount of body fat. So this article takes a decent amount of time to discuss future directions in which research could go. So the article discusses other research that show physical activity decreases maximal oxygen consumption. However, those who are physically active can reduce this decrease in maximal oxygen consumption by as much as 50%. Furthermore, the diabetes prevention programs show that even the oldest of old who have the high risk of developing type 2 diabetes can prevent it with diet and exercise. However, since diet was also modified, they can't just say that this effect was due to increasing physical activity. Thus, more research needs to be done into that. Studies also suggest that further research into different exercises will enhance whole body metabolism. And they bring up this idea of exercise snacking. So this is kind of like when you have a plate of freshly baked cookies sitting on the stove and you don't want your family to know that you're going to eat them all. So maybe you'll have one, you'll go watch TV, you'll come back, have another one, go study for a few hours, come back, have another cookie, go for a run, and before you know it, the whole plate is gone. So they want to take that approach into exercise. So you have a little bit of physical activity here, a little bit of physical activity there, and throughout your day, just small chunks that you're breaking off. So this is supposed to increase glucose homeostasis by altering aerobic and resistance training while you're doing your exercise snacking. And it'll also optimize glucose control with the timing and intensity. So the timing would be how frequently you're doing your exercise snacking and to what extent you're going, like the intensity that you're doing to your exercise snacking. This article also wants a group to design specific exercise protocols to increase insulin sensitivity, increase mitochondrial function, increase muscle mass, and overall physical capacity. The greatest takeaway and my favorite line from this entire article was the final line, and it said, "The proverbial fountain of youth may just be down the street at your nearest neighborhood gym." Thank you.